All right. Hi, everyone. Let's get started here with our webinar today. My name's Leah, and welcome to our Ask Us Anything session about human factors in IVD development. For those of you who haven't joined us before, we'll be bringing in some of our experts to talk about your most pressing questions in real time. We have had a large number of questions already submitted. Thank you to those of you who have done that in advance. We have questions around human factors requirements for IVDs and CLIA waived products, how to make sure that you've got a great study design that will actually meet the FDA requirements, which can be quite challenging. And questions about the timing. When do we do a formative? When is it the right time to do a summative? And then just some more practical questions about coordinating studies across multiple sites and keeping moderators consistent and any kind of real world challenges. I've got two of our experts here with me today. They've been through the ringer with IVD and helped their clients to success for many, many years. They have a lot of experience between startup businesses, just getting their first diagnostic products off the ground, but also worked with large pharma companies to do some more complex or more broader reaching products. So first we have Artie, our senior human factors consultant, who has 7 years of medical device human factors expertise under her belt. She's managed nearly 100 projects, moderated over 1,000 sessions, and contributed to about 24 successful diagnostic test approvals. So she really knows how to integrate human factors into this IVD process. She has a deep expertise in 62366, HE75, and our FDA human factors guidelines. Our second expert today is Allie. She's a senior human factors consultant as well. She's passionate about creating healthcare products that are not just safe and effective, but also enjoyable to use. We probably all can relate to that. She's done a lot of task analysis and user research for new concepts in medical devices, created and managed use-related risk analysis, and planned and executed both formative and summative studies. She's actually a biomedical engineer from Cornell, and so she's also has a great design background, designing user instructions for point of care and at-home diagnostics. So thank you both for being our experts today.
Thank you for the introduction, Leah.
All right, you're welcome.
Thank you.
All right, so next we'll go through how this session will work. Again, thank you for everybody who has already submitted your questions. We've already pre-prepared some slides that help answer those questions. However, feel free as participants to hop in and ask questions on the side using the chat feature or the Q&A feature. And then just so you all know, we will be recording today's session, so we'll be sending that out to those of you tomorrow who provided your email addresses. And if there is a question that we think might have some confidentiality to it, or you need to ask some more probing questions, feel free to reach out to us after the fact, and we're happy to hop on a call with you to go into some more detail about your particular situation. So let's get started. I'll hand it off to Artie to start off with the first question that we received.
Yeah, thank you all for joining. So we'll just hop straight into the questions and start answering them. But please feel free to, if there's any other additional follow-up questions or more detail that you'd like to ask about what we're talking about, please feel free to add in questions on the chat and Leah can interrupt us. So jumping straight in with our very first question. The question we got was, what are the key human factors requirements from the FDA, especially for IVDs and CLIA waive products? So there are a lot of different regulations, guidance, standards that typically, guide how we approach human factors for all medical devices. So IVDs falling under that category, most of the general guidance kind of dictates how we approach human factors for IVDs, and it's not too different from what we'll see for any kind of medical device or what factors typically govern those. Of course, with IVDs, like with any product, depending on the period of time, depending on the kind of product, the FDA does release specific regulations that are product specific or time specific. Great example with IVDs is, of course, during the pandemic when the FDA was issuing EUA authorizations for a lot of COVID tests. There were a lot of templates that were issued specifically for those that kind of guided specifically for that kind of product what the agency wanted to see. So it really depends on yours, like product, but in general, most of the guidance does come from FDA's applying human factors and usability engineering to medical devices. In terms of key factors that do guide how we approach human factors, the biggest factor, like in all medical devices, is always risk, the risks that are associated with use of your product. What are the levels of it? What are the harms that can come from it? Kind of dictates how much human factors needs to be done, the extent of it, how many or which tasks need to be covered in your human factor studies. Apart from that, there are other factors like users and use environments. So typically, for instance, the less trained your users are, the less professional they are, the more the agency expects to see human factors be a front-facing requirement and something that they expect to see data for. So for instance, if you have lay users, if you have over-the-counter IVDs, those are factors that will lead the agency to ask you to do more human factors work. Likewise, product design, depending on, for instance, with IVDs, if you have very complex result interpretation or it requires a lot more user work to be able to interpret, like for instance, if you have a lateral flow assay where you have to interpret or visualize lines, those are things where the FDA might expect to see additional work on that end of the workflow. And of course, there's the aspect of novelty. So for instance, if a product is new to the market, something that is very unique, something that's not been seen, and again, you have users who are untrained and aren't used to seeing that kind of product, that's where the FDA will put a higher emphasis. Again, using the COVID example initially, for those who've seen how the regulations have changed over time, when the pandemic first started and COVID tests started coming out, the FDA was requiring studies to have upwards of 100 users in the validation stage to kind of show that the product is safe and effective and people are able to do it correctly. But over time, as the populace became more trained and more comfortable, and the FDA was also more comfortable with the real world data that they were seeing, they dropped those requirements to 30 and 15, more in alignment with other medical device requirements or guidance that they have out there, to the point where even now they collect data, as part of clinical studies and so on, and don't have as much emphasis because now they're quite comfortable with it. Likewise, for other OTC products I've seen that are de novo or very unique to the market, I've seen the FDA come back and say, no, we want you to do more than what's the typical requirement over there. Ali, I don't know if you have anything that you'd like to add as well in terms of other factors that you've seen drive human factors requirements for IVDs.
Yeah, the other thing I'll mention is just, I know the question specifically called out CLIA waiver devices, and there is a specific kind of CLIA waiver guidance that can also be helpful if you are working on a CLIA waiver device. And that guidance specifically calls out the device being simple and having minimal risk for erroneous results. So those tie into the things that Artie already talked about kind of within product design and risk. But just wanted to call out that guidance as well, since I know CLIA waiver is an area of interest.
Yeah, great addition. Thanks, Ali. Okay, so I'll go ahead and move on to our next question.
All right, so I'll go ahead and kick us off for this
Ali.
one. So the next question is, how do you ensure the study design meets FDA requirements, and how do you define and identify critical tasks in your evaluations? So moving from kind of general HF requirements to validation requirements. So the HF guidance from the FDA kind of calls out 4 different areas to ensure to focus on for validation. So first is the users. We need to make sure that the participants that are in your evaluation are representative of the intended users of the device. And then critical tasks, which was called out in the question, I'll go into a little more detail in a second. But essentially, all the critical tasks identified for the device should be performed in the validation test. And then the user interface should represent the final design. So that's not only the device design, it's also any labeling, any accessories. So for IVDs, if there's like sample collection materials, all of those should be final. They should be in the final packaging with final labeling. The actual like box and that type of packaging should also be final. Sometimes that gets overlooked as part of the user interface, but especially for like OTC IVDs, the unboxing experience and seeing if users can locate the correct materials at the right times is an important factor. So all of that should be final or production equivalent. And then lastly, the test conditions should represent the actual conditions of use. So a big part of that is the study environment. The environment should represent kind of the intended use environment. So if it's a home use device, making sure it's not in like a conference room with really bright lighting that's not actually representative of what a home would look like, kind of spatial constraints, any other environmental characteristics that could kind of impact the interaction with the device, that should all be representative. And then the question also asks specifically about how to identify critical tasks. So I just pulled in the FDA's definition of a critical task just as a reminder, or for those who haven't seen it. So the FDA defines a critical task as a user task if performed incorrectly or not performed at all would cause serious harm to the patient. And it's important for especially IVDs to note that harm can also include compromised medical care. So for IVDs specifically, a lot of the times there's not those kind of risks like electrocution or cuts or things that might happen as you're actually taking the test. But things like an inaccurate test result or a delay in treatment, those can be big risks for IVDs, and that can kind of lead to compromised medical care. So those are important things to consider when you're kind of rating your tasks and determining what is critical and not. And then as far as how do you actually determine which tasks are critical? So the criticality of tasks is determined based on the severity level that's dedicated to it. So every task you should have a URRA, which is a use-related risk analysis that goes through kind of every task and what could go wrong at every task. Each of those possible use errors should be tied to a severity. So that's kind of what's the worst case that could happen if someone did that step incorrectly or did something that you're not expecting but is a foreseeable potential use error. If that's ranked at whatever you're setting as serious harm. So I highlighted an example from 14.971 that just goes through potential different severity levels, definitions. If you're saying serious or major is considered serious harm, basically anything with that or above should be marked as a critical task, and that should be included in your HF validation.
Yeah, and I think, Ali's mentioned a couple of really great examples of what we would consider serious harm when it comes to IVDs. And I think it really depends on, what condition you're diagnosing. For instance, if you're looking at misdiagnosis as a harm, it really depends on what you're misdiagnosing. Like the impacts of misdiagnosing, like a false negative or a false positive for a flu test might be very different than if you're talking about an HIV test or an Ebola test or something like that. And so you have to think about what's the kind of impact of a false negative, and that sort of determines whether something might be considered a serious harm. Another thing is, especially with IVDs, there's a huge aspect of sample collection. And so again, depending on what kind of condition you're diagnosing, the possibility of local area contamination is something that could contribute to serious harm. For instance, if you have a blood test or nasal swabs or any other kinds of swabs, if it sort of contaminates the table area and that's not inadequately cleaned later, can that sample then, assuming worst case scenario, somebody is actually positive for that condition and that sample is left and not cleaned off, could that later cause somebody else to be infected? What are the repercussions of that? Those are things that have to be thought of when it comes to IVDs and thinking about what really your serious harms are.
Yeah, and in addition to kind of incorrect results, also considering the risks of invalid results. So for especially like OTC IVDs, people are likely not buying like multiple tests, especially if depending on the cost. So if they do get an invalid result, that could potentially lead to a delay in treatment, especially if they need to go out and get another test. And for things like COVID, where Paxlovid and other treatment options have kind of a time window where it's more effective than others, that's important to consider as a risk as well if they were to receive an invalid result and that were to lead to a delay in treatment, which it likely would if that has any serious harm or any impact to the quality of medical care that they receive.
Yeah, and it's something I think like, that's a great point you bring up in terms of invalid results, because that's something that can also have a huge effect on your clinical trial as well. So being able to figure that out at the human factors validation stage, or even better, at the formative stage, is ideal so that you can kind of resolve and figure those things out before you have to report on a high invalid rate in your clinical trial. Are you ready to move to the next question?
Yep, I think so.
Okay. Cool. So our next question that we got was, how do you determine when a formative versus a summative usability study is needed? Which is a great question, and I think comes up a lot for all kinds of medical devices. So in general, here are some key aspects that we've got, listed out on formative studies versus human factors validation studies. And really the key difference is that the intention, the purpose, the stage at which you're doing these studies are quite different. So formative studies, as the name indicates, are done earlier on in the product development process when you're still iterating, refining, and designing and locking it in. And so the main intention is to uncover those usability studies and make changes to the device, identifying serious users, things that could happen and sort of contribute, like some of the things that you'll find in the formative usability studies will also contribute to your risk analysis and keep iteratively adding and refining your risk analysis so that you're making sure to capture any things that could happen based on what you've seen when users interact with it, and really just kind of explore device safety and usability with that priority on critical tasks. And the intention over there is, all errors that you're discovering, you kind of want to discover issues at the formative stage because those are things that you're then able to kind of fix or capture in your risk or really sort of explore and sort out before you have to validate. The validation study, on the other hand, is really done at the end of the development cycle and is something that you only ideally have to want to do once. And at that point, really, you don't want to have to discover any new use errors. Those are things that you would ideally have figured out earlier on in the process. And at this point, you're just showing that, hey, see, we've designed all these things out. So at this point, you're just looking to confirm that whatever risk control measures you've put in place are effective at reducing the risk, that confirming that you are meeting those FDA guidelines for human factors validation, and this is the data that you will submit to the FDA. So that's another key difference, is that formative usability data kind of contributes it, indirectly to the product development and just helping to refine the product, versus your validation study is really the data that you're going to package and present to the FDA to show the human factors. A lot of what you find out at the formative stage just goes inherently into the product design rather than really having to present that data directly front and center to the agency. And so that's why I say, like, that's kind of the stage where you want to discover errors, because then you can fix them, and what you're showing to the FDA later on in validation is much more clean and better, and you don't have the kinds of problems that you might have seen earlier. So like I said, the goals are really to learn the UI's strengths and opportunities for improvement at the formative stage, versus at the validation stage, you're really looking to prove that there are no issues, that the product is safe and effective, whether or not in the current form. The other thing is that because of the intentions of it, because of one being presented to the FDA formally versus the other one just indirectly contributing, the methodology tends to be more flexible for formative studies rather than summative studies. So for formative studies, you have your protocol, you have your structure that you're following, but there is more leniency in being able to kind of color outside the lines a little bit. You can have dialogues with participants, help to ask follow-up questions while the task is happening or after, because really the intention is, it's okay if this isn't exactly 100% representative. Our main intention is to really understand what are the issues, why are they happening, how can we fix it, and have that fluid conversation with the participant and just kind of drill down in it. And even ask them to do things like thinking aloud during the process to help them help you understand what their thought process is. Versus in the validation studies, there's a lot more onus to make sure that the structure of it is as representative of real life use as possible. The validation study is really intended to almost test out, worst case scenario at the minimum level of training, at the minimum level of guidance, at the greatest variety in terms of background of users, in terms of their experience and so on. How well can they perform with your product? And so when you're looking at the methodology of it, the scripts, validation protocols, and documentation tends to be a lot more clear cut and rigid. The script has to be followed very specifically, the order of tasks, and there is a lot of focus on not biasing participants by giving them information before they have figured it out themselves, or being able to have like a very clear evaluation where you aren't contributing any bias to it. Yeah. What have you seen, Ellie, in terms of, formative versus validation studies?
Yeah, I think you covered the differences very well. As far as formative studies, I also want to point out that these can kind of be used at a bunch of different stages in development. So as well as just kind of finding the use errors with the existing design, you can also use them to kind of guide your design and hopefully make a more usable product. So for example, for IVDs, if there's like a sample collection process where they need to add the sample to like a dilution buffer or something, there's some sort of sample manipulation happening, you can kind of use formative studies to figure out what is the easiest way for lay users, for example, to do that. Should they use like a swab and a pipette? Should they use a dropper tube? Like there's so many different materials you can use for those steps. And a lot of them are a lot more user-friendly and less risky than others as far as use errors. So you can kind of test out different materials and workflows and things like that in formative studies and help guide your design, as well as just testing kind of the design that you think might be the best. It's helpful to hear from users kind of throughout, and then you can really design it for them.
Yeah, I think that's a great point, because I think you've hit on one of the specific aspects that I haven't really covered, which is size. Formative studies tend to be much smaller in size than validation. The typical guidance is around 5 to 8 users per user group for formative studies versus 15 to 18 per user group for validation. But again, with formative studies, it's a lot more flexible. Even if you want to have 3 rounds of formative, each having only 3 users per user group, that's totally fine. The intention is for these to be quick and dirty. I know we have a question later on in our deck about flex studies, but that's another thing where it's like when you start to have human factors throughout your process and you start to collect that data and see what issues are popping up, it can also help you to plan out other activities, like things that, to be aware of that will pop up in your clinical trial, or things that you need, okay, we need to include these as among our flex studies. So there's a lot of other intentions and things that like formative studies can help with compared to validation studies, which is just kind of like a one and done and proving something is great. Yeah. Okay. Moving on to our next question. So this is about point of care IVDs. So the question was, when performing a point of care IVD assay, are there risks to an FDA submission to have a clinical testing site run a usability study prior to the method comparison study? It's a great question. I know with point of care, like CLIA waived IVDs and so on, this is something that we often see happening, is using the clinical sites to run the usability study prior to the method comparison. So there are a lot of considerations with these. They absolutely happen, and I think sometimes they are very useful, because when you are already planning to run a clinical study at a lot of different diverse sites and a lot of different socioeconomic areas, it's really great because then you can really capture that demographic variety as well. But combining studies in any format can always be tricky if either study creates bias for the other or affects the purity. So some things to consider with that approach is just having a lot more attention paid out on how separately or how independent you are keeping each study and each protocol, making sure that there's no bias. I think specifically with human factors validation studies, there's a lot of attention given to making sure that, you're not biasing the users by giving them information that they wouldn't have in a worst case scenario before running the test, or even asking them questions in a way that leads them or helps them to give a positive answer versus a negative answer, or really be able to honestly give their feedback in terms of issues. So some issues to consider is one of the other things with running it at a site. When it's a point of care and you have your clinic users as the intended users, it's a lot easier because the environment is representative and it is, there's a lot of different aspects of it, the space, the time constraints, and so on, which are representative of what you want to test. And so that's great. Sometimes you also see for, not this question, but for OTC, those are also being run at clinical trials. And those are things where you might tend to have a little bit of a reduced ability to test out those human factors considerations because you are essentially running a human factor study at a clinical site and that sort of environment rather than an in-home or in their living room or dining room, or kind of simulating that environment to the extent possible. Another thing to consider for any sites or any kinds of studies that are run at clinical trial sites is who's able to moderate those studies, who's able to collect data. With human factor studies in general, there's always sort of two prongs of data that you collect. You collect more independent, objective data from an impartial observer, as well as the more subjective data that you collect from the user or the participant themselves. And sometimes when you have at these big sites and there's a lot of moving parts, you might not be able to collect as detailed data from that objective end of things, of having a trained moderator who's able to observe the whole process and write down all their notes and the nitty gritty of things they saw where participants seem to hesitate or they're not looking at the right place in the manual or the quick reference guide. So just thinking about those issues that might crop up and being able to mitigate your design to ensure that those are kind of included in the study so that you can sufficiently establish root cause for the issues that you mentioned, and can then be able to talk about residual risk and identify whether or not further risk mitigations are required. The other thing I've already covered in talking about, your clinical study SIV, is there anything about the way that you're introducing it to the site staff or your operators that might unintentionally create bias for the human factors evaluations, or even vice versa, if there's something about the design of the human factors evaluation that once they do that for the first time and then they go forward and do it in the clinical trial, there's something about the design that needs to be different and causes them to make more mistakes. You'll have more deviations and things. Just things to think about. And also thinking about when you do have, let's say, 20 different sites running your clinical trial, are there inconsistencies between the setup or the evaluation at multiple sites? And on the flip side, if you're doing it at just a single site, are you collecting enough variety in terms of insights or being able to recruit diverse enough users in a separate study versus having only a limited variety in terms of intended use operators when you're just recruiting whoever the clinical site staff are? So suggestions to consider over there, just, designing studies as independently as possible, keeping, having an extra micro look at the design of the studies, the protocols, making sure that they are as independent, as rigid, as defined and laid out clearly as possible to make sure that you are maximizing the quality of the data that you are collecting, and then also looking at how these designs will impact, doing dry runs at the site and just seeing how that impacts how people perform before you really, run down the road and end up with data that is something that you'll need to put in deviations and so on. Another thing that I typically see with these kinds of things is when the human factors portion is also sort of added in or grandfathered into the clinical trials protocol, and sometimes that can work, but what that then causes is that there's a lot of aspects of human factors that aren't quite laid out in as much detail in the clinical trials protocol in terms of considerations of defining your intended users, environment, scripts, all those things. So making sure to have a separate human factors protocol that takes all those things into consideration and utilizing trained experts to collect and analyze the data to ensure their robustness, because you really want to be able to collect that objective data and kind of maximize what value you're getting from being able to watch these participants, see what they're doing, and use that to enhance whatever feedback that the users might give. Anything you'd like to add, Ali?
Yeah, I'll touch on a couple things. I think you touched on both of these, but just to re-emphasize, I think just thinking about the biases for each of the studies is important. And I know for CLIA waiver and especially like OTC products, things like that, the untrained users is a very important aspect. And it can be seen as some sort of training if it's not, even if it's not a formal training, if they use it prior to the other study. So they're kind of using it more maybe than they would in a typical environment, and you want to make sure that's not introducing training that's maybe informal but is still considered training by the FDA. And then the other thing is that, yeah, human factors data in general is different. So it is important to at least have some consideration of what type of data is needed from a human factors study, because it's not just what have we observed that people did incorrectly. You also need to understand kind of why that happened, and if that's what the residual risk of that is in real life. So it's not just observing what went wrong. You need to also have people understand how to get all of the data that's required, which includes what happened and why it happened.
Yeah, and I think that's a great point. That's making me think of one of the issues I've seen actually happen in a clinical study is we had the FDA kind of come back and throw out all the human factors data that was collected as part of a clinical study because the questionnaire that was designed and presented to users was too biasing in the sense that the way that the question was phrased, they were essentially statements that asked, what their experience was in different steps, but the way that the question was phrased seemed extra positive, and it led users to kind of were more likely to give a positive response or just say, yeah, it was great, rather than saying, no, I had these issues. And so the FDA was like, no, we can't use this. You have to have more neutral statements that people can kind of give their feedback on, rather than being able to just agree with a statement that's leaning positive already. So there are lots of these kinds of nuances that the FDA likes to think about, especially when you're looking at these untrained users with CLIA waived tests or even OTC tests.
Hey, Artie, we do have a question from the audience, if you have time for that. Question is,
Yeah, absolutely.
Is there any leverage of an IVD platform implemented as an LTD for human factors in the transition to FDA submittal for moderately complex clearance?
That's a great question. I think LDTs and moderately complex tests they're always, I think that's something that's being explored more and more now that the regulations have kind of changed for LDTs. And I think it really depends, and we like to joke that for all human factors answers, we always begin with it depends, but that's really the case, that every product is slightly different. So it would really depend on that specific, what that test is diagnosing, what that process looks like, how much user interaction there is, and how much power does the user have to be able to, create serious harm by making a user error. The other thing over there is, like I said, the FDA tends to pay more and more emphasis or more and more attention to human factors the less trained users are. So for something like moderately complex, you're going to end up having less scrutiny from the agency compared to something being CLIA waived or something that is on market or as an OTC, for instance, because those types of users tend to be more trained. They do have like lab training. They are higher level users compared to, your lay users who are doing tests at home. So it really depends, I think, on for that user group, how complex the product is and how many opportunities there are for use errors and risks. Hope that answers the question. I don't know if you have anything you would like to add, Ali?
No, I think you covered it. Yeah, it kind of all goes back to the key human factors requirements that I already mentioned earlier, which is a lot around risk. So it's hard to say for a general statement, but just looking at the risk, kind of the higher the risk level, which includes things like less training, the more FDA scrutiny that it would receive.
Yeah. And I think in general, like when we look at human factors validation, of course, when you're talking about a CLIA waived or an OTC, there isn't really any training. So when you're conducting the human factors validation, you have to account for that and not give your participants training. But the FDA wants the human factors validation to be as representative of the worst case scenario that will realistically happen. So for higher level medical devices where the company is providing training or something by default at a basic level, every user who ever uses this product for the first time will have had this training. That's something that also gets built into the human factors validation study where we have to simulate that training. So all that to say, if there is training that you are providing as a minimum before performing this test for the very first time, that's additional justification that you can use to show, a reduced need for human factors validation or be able to build that into your study and have, better outcomes as a result because you've kind of accounted for what you're providing in real life. All right, moving on to the next slide, next question.
Yeah, so the next question is, how do you ensure consistency in moderator-led sessions across multiple sites? This kind of ties back into if you are implementing an HF validation in a clinical trial, you're going to have multiple sites most likely. So a couple tips just as far as ensuring consistency. So the most important thing is to have a very clear moderator guide. So this should be kind of a detailed script for all the moderators just to ensure the language is very consistent and that non-leading wording is used. The script should include kind of everything from the introduction to the session, including also the prompts that are going to be provided to the participants as far as what tasks you're asking them to do, and also any knowledge questions you're asking them, as well as kind of those follow-up questions to get subjective feedback. The script should include all of that so that the same wording is used across sites. Additionally, the moderators should all be trained. This goes back to having someone who understands kind of what human factors is and what type of data you're trying to get. Especially bias training is something that can really help make sure that human biases aren't affecting the usability data and is not always something thought about. And then also just making sure all the moderators are trained on the study protocol and are following the moderator guide. And then ensuring the data collection is done consistently across sites. So having the same data collection sheets and also making sure that how people are or moderators are rating task performance is defined so that that is done consistently across sites as well. And then finally, the test environment should be pretty standard and the materials that are available to the participants. So that's not just like the device and instructions and that stuff itself, but things like a sink or hand sanitizer, washing hands is an important aspect, making sure all of those different study materials are also consistent across sites.
Yeah, I agree. That's all great points. And I think this also speaks to some of the points I mentioned earlier in terms of when you are trying to conduct a human factor study at a clinical site, just making sure that you have a protocol that really lays out all of these things as scripts, as, as rigidly or as clearly as possible so that you can keep that consistency across different sites and you sort of reduce the possibility of variation because everything is just really scripted out and well choreographed across all your sites. Yeah. Okay. We'll move it to the next question. And I know we are running closer to time, so just in case we aren't able to get to the specific question that you have submitted, please feel free to reach out to us. I know we have some email addresses at the end of our slides where you can reach out to us and we can set up follow-up calls and discuss your specific question with additional details, as Leah mentioned. So our next question is asking about OTC studies where the clinical study coordinators are acting as moderators for each subject. Are there any good practices to standardize how steps are observed and scored between sites and coordinators? This is somewhat similar to some of the questions that we've covered in terms of consistency across sites and just thinking about combining or running a human factors study and a clinical study altogether. So I won't go over those things that we've already mentioned, but some additional things to consider when we're talking about OTC human factors data. Like I mentioned, when it comes to a CLIA waived product, if your intended users are your clinic's site staff, you are kind of representing that intended use environment already at your clinical site. And so it's a little bit easier, a little bit better to kind of define and justify versus with an OTC product, there has to be additional attention paid to how well you are simulating that intended use environment to what they'll actually be using. So some of the points that Ali's mentioned, for instance, is the the lighting of a space and making sure that it's not too bright compared to what it's going to be in real life. So thinking about all those different aspects. Another thing, like I've mentioned, a really great human factor study will always combine objective as well as subjective data. So making sure not to rely on self-reported usability. I already mentioned that one example I've seen of the FDA just kind of throwing out basically all the usability data from a clinical trial because it was all self-reported and they didn't like the way that the questions were phrased. So thinking about that, and there's always when it comes to users and participants, people have a lot of barrier, a lot of hesitation to point out issues in the product, and they'll tend to blame themselves. So self-reporting, relying on self-reported usability can lead to just a lot of issues not being uncovered. Another thing to think about when you do have a lot of studies when you do have a lot of sites is larger study teams can lead to inconsistency. So standardizing that methodology like Ali laid out, the different factors that you really want to call out is key to making sure that the data is robust. And consider the balance between quality versus quantity. If you try to collect human factors data from every single one of your clinical trials participants, you're going to end up with, you could end up with like 1,000 data points, but how deep or how high level can you really have when you're collecting data from that many participants versus being able to run a smaller study and focus more and do more intensive work with each of those participants in terms of collecting detailed observations and how they interacted with it and have more to put into your root cause analysis and residual risk analysis. A thing to definitely keep in mind when you aren't recruiting or including every one of your clinical site participants in your usability study is making sure that you don't cherry pick your participants. So a huge emphasis especially when it comes to OTCs and especially when it is that all your intended users are essentially like the United States population, there's a lot of emphasis given by the FDA in trying to make that as representative or even representative of users who have lower educational or socioeconomic backgrounds. And so making sure that your participants that you're including in your clinical trial, you're not cherry picking them to be higher level participants, people who are performing better, or people who lean towards any one demographic group and overrepresent that compared to, what the United States Census might represent in terms of that demographic subpopulation. Okay. Ali, did you have anything to add, or should we go ahead and move to the next question so we can try to get through all of them?
Yeah, we can skip around a little, try to touch on some topics we haven't talked about yet.
Yeah, agreed. So moving on, there's a question about factors to consider when we're recruiting for an OTC validation study. I'll go through these like really quickly. These are just some the typical characteristics that the FDA likes to see in terms of looking at your demographics and kind of defining who your intended users are, making sure to represent ages, literacy levels, vision, any physical dexterity and motor skill levels, socioeconomic and educational backgrounds. The only thing I wanted to call out for OTC IVDs specifically, especially when your intended users are, again, it could be anything like, let's say, a COVID test or flu test, and really there's no specific subpopulation that it's geared towards, it's even more important to demonstrate that worst case scenario. The United States Census is always a really good background to use when you're looking at how much you want to relatively recruit your different subpopulations in terms of races, ages, education levels, and so on. Even trying to overrepresent some minorities is really helpful to kind of show that you've kind of tested that worst case scenario. The other thing is the novelty of a device can impact how large a sample size the FDA expects to see. Like I've mentioned, typically the standard guidance is 15 users per user group, but I've definitely seen for newer novel products, the FDA coming back and saying, no, actually, we want you to do 80. We want you to do 100 because this is really new and we need to see how untrained users kind of react to this very new product. So thinking about those things and how to represent and collect data really show that you are demonstrating the worst case scenario. I think this is a question now on data, Ellie.
Yeah, do you want to actually just go ahead and skip to the flex studies? I think we've talked about this a little bit, so I want to touch on some other subjects. So there is a question about how flex studies tie into human factors and use-related risk. So you go to the next slide. There's kind of two primary benefits for HF, kind of how you can use flex studies within your HF process. The first is de-risking the HF validation testing, and the second is supporting residual risk analysis that happens after the HF validation testing has completed. So I'll go ahead and talk a little bit about de-risking the HF validation. So you should expect variation as far as how users are going to use your test, especially if it's an OTC product. There's no training. It's expected there's going to be some variation either intentionally or by mistake from users. So flex testing can kind of help understand what amount of variation won't impact the results versus what is actually considered a use error, what will impact the test results. So we wanted to give an example of how you can kind of use flex tests to figure out where that line is, and then use that to set your acceptance criteria for human factors validation, which should limit kind of the non-representative use errors that need to be reported if you didn't necessarily know where that line was. So for this example, we're going to use a swab in the nostrils. I think we've all kind of used tests like that with the COVID testing. So say the instructions tell the user to swab both nostrils 5 times. The instructions may say that just to kind of get sufficient sample, but in a lot of cases, less than that may give sufficient sample for the test to run properly. So understanding kind of what is the actual minimum amount of sample collection required for a result to be accurate. Say you ran flex testing, kind of doing different methods of swabbing your nose, and found that 4 total rotations, either all in one nostril, 2 in each, any sort of variation of that provided sufficient sample for the test. With that, you can set your acceptance criteria for your validation test. So rather than saying a user is successful for the sample collection if they swab both nostrils at least 5 times, you can set that to they swab their nostrils at least 4 times total. So just to show kind of how that impacts the use errors that need to be reported, you'll see kind of if there's no flex tests and you're following the instructions, basically only people who swab both nostrils for 5 rotations will be marked as successful in the validation test. Everyone who does something different, so say someone just miscounts and does 4 rotations in one nostril and 5 in the other, you'll have to investigate what's the root cause of that, what is the residual risk of that, versus if you've done a flex test, you kind of understand what the risk is of that, and that if there is no risk, then you don't need to go through that entire process for the validation test. So if you did run your flex test before and found that 4 rotations was sufficient, kind of all these different possible combinations of user performance in the validation test would be considered successful. So they could follow the instructions and swab both nostrils 5 times. They could swab one nostril, kind of swab both nostrils twice. Just all the different types of sample collection that as the developer will lead to accurate results, but aren't necessarily what's said in the instructions. So it just gives room for some user variability that won't impact the test results. And that can help your validation study be a lot cleaner and lower risk.
Yeah, I can tell you from every single study that I've done, anytime that there is a number of times that someone's supposed to do this, or even when there's a certain number of seconds that's prescribed, I think seconds, for instance, is where there tends to be so much variability that we see in studies because everybody counts at different paces. So, I mean, I'm sure at some point everybody here has probably run a COVID test and they have this thing where you're supposed to swab 5 times for 15 seconds. And I've seen the whole spectrum in terms of how people count 15 seconds. We would have like timers and we would kind of be evaluating. And I've seen people do it for 5 seconds. I've seen people do it for 10 seconds, people doing it even for like 20 seconds because that's how slowly they're counting. So making sure that when there is those like time components or things that we want to do, or even like waiting 30 minutes for a test result, not people, especially when it's something like at home, they're not going to set a timer. They're not going to be super rigid with being able to count those things. So you can expect to see a lot of variation whenever there's that kind of like number prescribed.
Yeah, and then the second,
sorry, speaking of COVID-19 tests, it might be a good time to answer a question from the audience here. How has the widespread use of the COVID-19 test impacted lay users' approach to IVDs? I think most people might not have seen them until the recent years, right?
Yeah,
It's a great question.
That's
Do you
an
want to
interesting
start, Ellie?
question. Yeah, I can start off. I think that it's interesting because I do think that for a lot of people, COVID tests were kind of the first at-home IVD that they had experience with, maybe outside of like pregnancy tests or other things like that, but not everyone had seen them. And I think COVID was definitely kind of the widest and quickest release of kind of in vitro diagnostics in the home. So I do think that kind of shaped what we call the user's mental model, which is what their expectations are of how a device works. So at least from what I've seen kind of post-COVID, when people approach a new test, even if it's testing completely different pathogens or is a completely different technology, they'll often approach it kind of with the mindset of what they know about COVID tests. So that brings both kind of expectations as far as how it works, which if it works differently can cause use issues. And it also, one interesting thing I've seen is it does kind of have some mistrust with it. So with a lot of the COVID tests, there was kind of this period where people were like not trusting the results, especially if it was kind of ambiguous or they were getting mixed results, taking multiple tests. There was kind of a doubt as far as what is the accuracy of this test. And I've seen that kind of translate into new tests that are being developed. One of the first questions people ask is, how accurate is this? How do I know if I can trust my results? And a lot of people sometimes will even say, I would just take another one just to make sure based on like past experience. So it is interesting just to think about kind of what were the strong points of COVID tests, what did people do really well, and then also what problems did it create and how do we address those in new diagnostic tests?
Yeah, and I think it's always the case when you do have like a population has limited experience with a specific kind of task or a specific kind of thing, that they're more likely to relate it to the one example or try to fit it into the box that they're familiar with. I think even when I was running a lot of COVID studies, I had a lot of people who, had previously taken pregnancy tests who would try to equate their experience or try to equate the steps of a COVID test with the steps of a pregnancy test because that was the only single example that they were aware of. So I think even when, people were running COVID tests initially, they did start to see those as well with other products like pregnancy tests impacting it. And I think like over time, as more and more OTC products reach the market and people get more and more familiar with a variety of different workflows, that will sort of ease up and there will be less of a fixed expectation when they come in to use your product. But I think you can look back at examples of how great developers have designed out some of those issues or risks by, creating a design that relies less on the user to interpret something or potentially make a use error. Even pregnancy tests these days, originally pregnancy tests were always like lateral flow assays where you had to interpret the number of lines, 1 line's negative, 2 lines positive. But these days you see a lot of digital pregnancy tests where it kind of takes that ambiguity out of it and you just rely on that digital readout, which is a lot more clear cut. So thinking about, okay, where is there possibilities for negative transfer and making sure to kind of design those out and keep those in mind.
Yeah, and I think one other thing that's actually a positive from the COVID test is people, especially lay users, do seem to feel more confident using at-home IVDs. So I know I've had people in the past in just formative and validation tests mention like, oh, a couple of years or like 5 years ago, I never would have been able to do this. Like this would have scared me so much to try to do at home. But now that I've gone through the COVID tests, I understand kind of the basics of how it works, or at least they have some idea of what it is and how it works. So they have more confidence going in, which can help as far as just usability in general and making sure that they feel confident going through the process and feel like they would use a test at home, whereas they may not have before.
Yeah, great. So I think we have one last question over here that we'll try to get through super quickly. So there was a question about being interested in the URRA guidance, interpretation, and CLIA waiver, and if it will say in effect. So,
yeah, so
Ellie,
I can kick us off here. So just to point out a couple of key differences between the two guidances that were mentioned in the question. So the URRA guidance, draft guidance that was recently published is targeted at drug-led and biologic-led products. So the whole guidance is kind of just a more formalized expectations of the FDA regarding URRAs, and it kind of extends the application of URRAs to drug-led and biologic-led products, in addition to kind of device-led products. So the CLIA waiver guidance, on the other hand, is applicable specifically to IVDs, so more device-led products, generally speaking. And in that guidance, there is some mention of risk analysis. So they mentioned to do a systemic and comprehensive risk analysis. And I think this question was touching on kind of if there's any maybe new information in the URRA guidance that would negate any information in the CLIA waiver guidance. But they're both kind of saying similar things, just with different specifics. So the URRA is something that's been used in human factors for device-led products, it's kind of a standard practice. So while the CLIA waiver doesn't mention URRA specifically, it is a good tool as far as doing a comprehensive use-related risk analysis. And the CLIA waiver guidance does kind of point out different operator errors and human factors that could lead to use error for consideration. And those kind of feed into the URRA. So those are things to consider as far as what can go wrong or what could a user do that's not as expected. So I guess in my mind, these are kind of complementary. They're not necessarily negating one versus the other. They say similar things and are kind of just providing ways to do use-related risk analysis. The new guidance just kind of sets out a more formalized process for drug-led and biologic-led products.
Great. And I think in general, it's sort of representative of a trend that we've seen over the last few years where in general for all medical devices, the FDA kind of wants to see that use-related risk framework rather than just seeing like a hazard analysis and so on. And so they really want to kind of have you put that in that framework of according to your use steps and being able to identify risks. And the more and more we see the agency changes and seeing the reviewers start to diversify. So you're not necessarily getting an IVD specific reviewer and kind of seeing those changes, keeping in mind what the broader medical device industry is seeing and what the general trend is in terms of the data that the FDA expects to see is going to be really important, and just try to follow that even if it's not necessarily called out specifically for CLIA waivers. So Okay. Time for wrap up. I know we've gone over a few minutes, but thank you all for sticking around and listening to us. I know that there's probably some questions that we went through quite quickly. So if there's additional follow-up questions that you would like to ask or more details that you would like to ask, please, we'll send up follow-up information or schedule time to address your specific needs. So if you have any questions, you can contact us at info@ClariMed. We are going to be having many more Ask Us Anything webinars on different topics. And this is the, I think, second of an ongoing series with different experts to address different medical device topics. You're already on our list to receive invitations. And if there are specific topics you'd like us to cover in future webinars, please do let us know. Reach out to us at that email address. Here's some more contact information from me and Ellie directly if you would like to reach out to us with any follow-up questions. Thank you so much for your time. I'll leave up the contact information here. Leah, Ellie, anything you'd like to add?
Oh, thanks everyone for attending. Thanks both of you for your expert advice and sharing of stories. And we hope to hear any feedback that or additional questions that come out of this from attendees. Thanks.
Yeah, thank you, everybody.
Thanks all.